You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. My guest is Laura Towart. She's the CEO and founder of My Personal Therapeutics. So, Laura, thank you for coming. How you doing?
Thank you very much. Thanks for hosting me.
Yeah. So, tell me, if you would, what's the premise of the company? What's a little bit about your background, and how did you come to create this company?
Sure. Well, I'm a biotech kind of aficionado. Personalized medicine is one of my main passions, and something I've been working on for quite a while since my PhD studies. I have a history in entrepreneurship. I started another company called Cellmatics, which is a New York based biotech company pursuing personalized medicine in women's health. And I founded that company about 10 years ago. But for the last 7 or so years, I've been working with Mount Sinai Medical Center in New York, and very interested in their technology that they've pioneered called the personal discovery process. It's really an interesting technology because it has very widespread applications across all genetic diseases. And the way that the technology works is, the technology is mainly used for cancer, or so far it's been mainly used for cancer. The way that it works is we would take a patient's tumor biopsy, sequence it, kind of detect what are the main drivers of the tumor, the main genetic mutations driving tumorigenesis, and then we build them into fruit fly avatars, and then we do large-scale, high-throughput drug screening to identify new combinations of drugs for patients. And so so that's the company.
What is a fruit fly avatar?
So a fruit fly avatar is a fruit fly that's been genetically engineered to develop a tumor that's very similar to a patient's. And then we can use that fruit fly to do experiments on to discover the best fit drug treatments for a patient.
Oh, that's really interesting. How did you figure that concept out, or where did the concept come from?
So it came from the Icahn School of Medicine at Mount Sinai Medical Center. They developed it over about a decade, and it's the center that they've created around the technology involves oncologists, basic researchers, pathologists, it's genomicists, network analysis, data scientists, and so it's really a very large effort that's gone to making what seems a very simple idea of creating a fruit fly, but to make the technology applicable to humans.
Well, fruit flies develop much faster. So what happens if, I have a tumor with certain markers and we put it into a fruit fly? How fast will it develop?
So it develops really fast, and that's actually one of the greatest utilities of the fly. Other people are modeling making avatars in mice, but it takes a very long time for the mouse to develop. And also, you can't do high throughput screening in mice. With flies, they're small, they mature quickly, and they're also they don't require as much drugs, so that it's actually a lot cheaper. So we can do really high throughput screening in a really fast time.
So the in in vivo environment of a fruit fly, you're saying is what, somewhat similar enough to a human where you can test certain elements of a tumor?
Yes, so there are some limitations to the fly model. Some tumors are better modeled in flies. Other tumors might be better modeled in a system like a zebrafish or mouse. But for the flies, like colorectal cancer is very well modeled, lung cancer. A lot of other cancers, the tumor types that are difficult to model involve hormones. So the flies don't have hormonal systems. And so we could mimic them by engineering in hormone receptors, but it becomes a little bit less natural. And so the company right now is focused on modeling the tumors that are well modeled in flies, and then we'll maybe pursue some other more difficult modeling later.
It makes sense. I mean, it's like a very strangely creative way of doing it. What aspects? Well, first of all, what cancers are well modeled in flies, and then what aspects of the cancer can you model?
So colorectal and bowel cancer, GI cancers, lung, some sarcomas. Most cancers any epithelial based tumors. The difficult ones are really breast, prostate, anything that's involving hormones. Brain cancer is a little bit difficult, depending on what type of brain cancer it is. But that's something that's an interest of some of the scientists that are working on our team, and so we're doing research and development towards that aim now.
So specifically, what kinds of things can you model about the cancer? What can you look for?
So we basically take up to 15 genetic mutations, and so we're kind of unbiased. We go into looking at the tumor genome, and we do unbiased identification of mutations, which means that we're not just using a genetic panel of known oncogenes. And so that's really what the standard of care now is patients that are doing genomic profiling of their tumors. They're mainly looking at something like a Foundation Medicine 1 test where they're looking at known oncogenes, and they're saying, oh, okay, you're KRAS positive, you have this mutation, and then they use drugs to target KRAS. What we're doing is really looking at all of the genes that might be mutated and that seem to be playing a role in tumorigenesis. We're looking at definitely known oncogenes, but also genes that might be working with the oncogenes, and then other kind of passenger mutations, genes that are working in mutations that are working together, like kind of in clusters. And so we can model all of those into the fly, and then so that we get a tumor that's growing in the fly that's very genetically similar to that of the patient.
Well, what happens when you take tissue out of a person, do histology, pathology, look at the tissue, and you sequence the DNA there? Do genes turn off? Does it very quickly not resemble what it does inside the person's body? Is that why you have to do it inside an in vivo fly environment?
So people are doing these kind of patient-derived kind of xenografts where they take tumor tissue and then they culture it, and then they do kind of drug discovery or kind of looking at drug combinations that might work. Our argument against that is that you're really just taking the tumor and putting it on a dish, and so you're looking at it in isolation. You're not looking at it where you're taking away all of the ability to look at signaling, and so we would say that that's even more artificial. The fly enables us to look at the tumor as part of a whole animal model where we can look at signaling and interaction with blood vessels and things like that. So it becomes more natural.
Well, once you input the tumor into the fly, I would think there's a period of recognition and integration. I mean, I would bet there's tons of cell-to-cell communication that the tumor does with its microenvironment and with its host. So is there how soon after you implant it into a fly do you start looking at the tumor, and have you noticed that it changes from its earlier baseline significantly?
So we don't actually implant the tumor. What we do is when we identify the mutations, we create like a plasmid type construct And so we build the kind of the plasmid, and then it's basically micro injected into the pupa of a fly, and so the fly will grow up to develop the tumor, and so it's basically something that will grow up with the fly.
Well, as the fly grows up, I mean, how long does it take for it to grow up? Like one day, or how
Yeah, it's
long?
very fast. It's really fast, and we can control it based on temperature. So if the fly is in a warmer environment, they grow faster, and so we use temperature as a control, like to our advantage.
Well, how would you know how the tumor or the tumor cells or the material in the plasma, how do you know how much it changes in its new environment?
I don't understand.
I would think that, the action of the tumor, I mean, would dramatically change possibly in its new environment. The material, the genes would, I don't know. I would think that their use and they would change very dramatically. Do they not? Is it preserved? How well is it preserved in this new host?
Well, we're looking at trying to develop a tumor that's as genetically similar to the patient's. So, I mean, it's going to be a little bit different because it's not having all of the genes that are making up the tumor that are playing a part in the tumor and in the human, but it's kind of the best model that we can do. For example, if you're trying to recreate a tumor in a mouse, you could only take a handful of genetic mutations because just by nature of the way that you can genetically engineer mice. But for the fly, you could select more mutations, but it's still it's not going to be the same exact tumor. I mean, of course it's going to be different. You're not going to have the interplay of all of the mutations, but it's going to be kind of the best that we can do.
So what exactly are you putting into a plasmid structure and injecting?
So
all of the nuclear DNA of a tumor cell, or the entire cell itself, or what's getting injected?
It's basically it's like a promoter driven construct that has all of the mutations built in.
So it's just my lack of understanding. I'm sorry. So it's you're not injecting a cell. Are you injecting just some DNA, or like what are you actually injecting? Is it some DNA inside of a membrane, or what is it? I mean, can you describe it?
It's a plasmid. So okay. it's like a small DNA molecule that's we separate it and you could micro inject it. So it contains it contains
so
all
it's
of
almost
the.
It's almost like a virus, right? It's just like a membrane, I guess. It's a cell membrane with DNA material.
It's a bacterium.
Okay, but again, like physically, it looks like just like a membrane material with the person's DNA inside of it.
Exactly.
Part of their DNA.
But we don't actually put the patient's DNA. We genetically engineer what
okay.
Yeah, so we take the patient would have their tumor biopsied in sequence, and then they send us the sequence. So we don't receive any patient material at all. Our process is all kind of bioinformatic based, and then we create our own constructs for microinjection.
Oh, so they'll say, all right, we're looking at these 10 different genes, and each of them is what, like, I don't know, I'm just making this up, 10,000 base pairs long. So let's say that's what it was. What would the construct look like? How would you emulate that?
Well, there's a lot of. I'm not the person that does the kind of the construct building at the company.
Okay.
I'm not the chief scientist, but basically, like it has a lot of hairpin loops.
Okay, no problem. Yeah, I was just asking for the mechanics of it, but no worries. Okay, the concept still makes sense. Gotcha. So how far have you gotten with doing this? You tested it on flies, and what have you observed?
So Mount Sinai has an ongoing clinical trial, and so they've enrolled 23 patients, and they have treated 9 patients. And so one of the first patients that they have outcomes data on was published in Science Advances in April, and so that patient had KRAS positive colorectal cancer and was successfully treated with a novel drug combination that could have only been predicted by the flies. And so we've initiated commercialization, and so we're selling personalized treatment designed to patients now.
Oh, so the procedure would be that someone comes in and they have this treatment-resistant cancer of the right type. You create this fruit fly avatar, and then what do you start doing? Like rapid-fire testing as many different drugs as you can against that construct to see which one has an effect?
Exactly. We do, using robotics, we screen up to 1,500 FDA-approved drugs in combinations of up to 3. So, tens of thousands of drug combinations. And then we see which combination rescues the fly. And then we make human treatment recommendations.
Wow. How long does it take to see a positive effect if there's going to be one?
If there's going to be one, we see it quite rapidly. But the overall time that it takes to build the model and do the screening, it is quite long. If it's a tumor type that we haven't done before, it could take up to 5 months. So we recommend that if a patient is starting to develop resistance to their standard of care, they should start their model, start their PDP. And we're working to improve this time. Like, for example, Mount Sinai has done a lot of patients with colorectal cancer. So we're at kind of what we call a product called targeted personal discovery process. So we've become more intelligent with the patients that we've had already, like by the experience that we've had. And so using artificial intelligence and kind of deep learning techniques, we can rule out some drug combinations that we know that aren't going to work. And so thereby we need less flies. And so it's cheaper and faster. And then over time, we know that patients with similar tumor genomic profiles will respond to the same drug treatment recommendations. And so if an incoming patient is very similar to a patient we've already done, we don't have to build the fly. And so over time, it'll be a fully kind of big data product. And so that's what we're working towards now. And our big data product will be called Match.
So you'll build up your flybrary.
Yeah, well, I like that. No one's ever said that before. I like that, the flybrary.
Well, so these are already FDA approved drugs, but are they approved for another indication, or how do you know which ones to even look at?
That's a great question. So yes, we screen all of the drugs. We do unbiased screening. But one of the kind of pitfalls is that a drug might be not approved for that indication, and so then it might not be covered by insurance. So an oncologist could recommend treatment with a drug that's not approved for that indication, but insurance might not cover it. So that's something that we're working with pharmaceutical companies now to offer some of the an allocation of their drugs for compassionate use, so that if we see that a patient would benefit from their drug, but it wouldn't be covered, we ask them to donate some. And so Mount Sinai's had a lot of success with doing that for the patients in the clinical trial.
But if a drug works, for a certain type of resistant cancer, I mean, then the benefit would be that you could tell the drug maker, hey, it's worked. And then the drug maker could go apply for a patent or for FDA approval for that specific use.
Exactly. Yeah. So we actually would be helping the drug company, because we would be saying, hey, we found another use for your your drug. You should start applying towards having it approved for that indication, and therefore that would also then help us.
But you're like grandfathered in. Then that you're allowed to get like a reduced price or something, or free access to it in return for doing this.
We haven't had any conversations like that yet, but I think that's a good idea.
Well, yeah, you want benefit back and forth.
Sure. Well, I think our benefit would be that the patient also is able to use our drug treatment recommendation.
Right. Are you able to test drugs that you think structurally might have an effect but aren't even FDA approved? I mean, since they're in fruit flies, can you test anything or no?
So technically we can, but okay. we have.
But do you?
No, we do. There's a few reasons why we don't. One is because we're really busy trying to identify new combinations of drugs that we can use today. We do have a longer term interest in doing so, but that's something that Mount Sinai might be doing separately from us.
So I guess, okay, so it'd be easier if you use an FDA-approved drug because that drug should not have any cardiotoxicity or liver toxicity or anything. But if you just use anything off the shelf, even if it works, it may still have other problems that, because it hasn't cleared the FDA.
Yeah, and doctors wouldn't be able to recommend those drugs.
Right. I just didn't know if there's like an Ultrafast fast track. If you see something works, why not? I mean, I know it's a whole nother project, but
yeah,
the FDA allows this. Why not let them allow getting an ultra fast track to be able to be used?
Sure. No, I think I think first we have to prove. I guess we have to get more data that our fly based predictions are correct for everyone, including the FDA, to gain confidence in our platform.
Okay. Why does it take so long if it's a new construct? And why can it take up to 5 months? What eats up all that time?
So the main kind of time consuming part is when we micro inject the flies, and then we need the flies to grow up, and then it's difficult to get. Let's say if we make a fly model, if a patient has, let's say we make a 12 mutation model. You can't make one fly at once that has all 12 mutations. So you could make a fly that has, let's say, 6 and another fly that has another 6. And then you cross them until you get ones that have 12. And then you've got to grow up enough flies to, if we're going to screen, let's say, 400,000 avatars for a patient, we have to get that many flies. So it's basically a breeding process that takes a long time. So as we get smarter, it takes shorter because we don't need as many flies, and then until we need none. And so that's really the most time-consuming part. But to be honest, we quote that it would take 5 months for a tumor that we haven't done before. It might take much less, but we don't want to overpromise. For colorectal cancer and cancers that we've done, we'd say expect it to take up to 3 months. But if you have a higher probability of a patient having a tumor that we've already have a very similar one that we've done, and then therefore we can start with that drug combination, and if it works, it works, and if it doesn't, then we can kind of open it up to other combinations. But some patients might get lucky.
How do you so these flies grow up and they turn into regular flies and start flying around and stuff and interacting?
Yeah, the ones that we successfully treat. The other ones, all of the flies that we micro inject the mutations into, all of those will develop lethal tumors, and so they will all die pretty fast. And only the ones that we find that the right cocktail will survive.
But how do you tag the flies so that fly over there with the the long hair on its head, that's the one that has this mutation. Let's get it back and test it. I mean, if you have all these flies flying all over the place, are they in little isolated chambers each of them? Or?
They're in little chambers. They're in, we use a lot of different, depending on which round of screening it is. It can be something like a 96 well plate or something. It's kind of small. Everything is very micro.
You can't inject any of the flies and then breed them after that, right? Like, how do you breed the flies?
It's just standard fly breeding, putting them in the same kind of the same tube.
Okay, but you'll identify the two flies and then you say, all right, go hang out, go breed, and then you separate them. Yeah,
we can tag which flies have mutations. Some of them will be tagged with like green fluorescent protein, so that then we can look and see where it is. And so there's all different ways of tagging the different flies. Some of them you can make a curly wing versus a non-curly wing. So there's all different ways of tagging the flies.
Boy, if someone went out with you to eat at a restaurant and there was a fly there, I wonder what you would think. You'd probably be like, I don't know. You would think what no one else would think by seeing a fly, near the food.
Yeah,
wondering,
Yeah. Well, these flies actually, we're not really allowing them to fly around. They're really confined in their tubes. So they're and the ones that have cancer, they're not going anywhere.
Right. I know. I just wonder what you would think if you just saw a fly, out and about in the real world. It would probably make you think a lot more than someone else.
Yeah, now when I see a fly, I'm grateful because I think, wow, there's so many things that we've learned from the fly. The humble fly is, really helped advance human genomics.
That's cool. So these are what, fruit flies?
Yeah, they're Drosophila.
Langosters.
Yeah.
Okay. Well, very cool. So what's the future of these fly avatars? What new things are coming, or you're just busy testing? Like what's ahead for the company?
So what's ahead? So we're kind of working on some cool new collaborations. We're working with one of the largest diabetes centers in the Middle East, the Dasman Diabetes Institute in Kuwait, and we're actually making diabetic flies that also have cancer, and we're studying how diabetes changes the polypharmacology. And so it's interesting because the flies that are diabetic would receive completely different drug treatment recommendations. So that would indicate that people with diabetes should have different treatment recommendations. So it's a whole new field of kind of research for us. And then we're also able to model any genetic disease, and so particularly rare genetic diseases. And so we're going to start to work with some rare genetic disease societies to help identify patients that we might be able to help.
How do you treat a fly, by the way, when you give it a certain cancer? Does it drink the medicine, or do you just inject the medicine? Like, how do you treat it?
So the flies actually eat the medicine in their food.
Okay, eat it. Gotcha. Huh. Interesting. This is really interesting. Well, very good. So
thank you.
What's a goal of yours for the next few years? What do you hope to accomplish? Just more drug discovery, or there's like specific milestones that you're hoping to achieve?
We're really hoping to identify new drug combinations and really help find the right combinations for people and really educate the public on personalized medicine and concept and not every person. We really want to develop kind of rapid, affordable, personalized treatment for patients with cancer, patients with other rare genetic diseases. So we're starting with cancer, and then we'll start to look at some neurodegenerative diseases and other genetic diseases.
Okay, very good. Well, Laura, this has been like a super interesting call, and I really appreciate you coming on the podcast. What's the best way for people to, how can people ask questions and, get in touch with the company and learn more? What's the best way to do that?
Oh, sure. Well, please visit us at mypersonaltherapeutics.com, and I'm happy to share more information with anyone. My email is laura@mypersonaltherapeutics.com.
Okay. Well, Laura, thank you for coming. Appreciate it.
Thank you so much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.